Yeah, thank you. I see people like coming in still, and so it's thanks for the organizer giving me this opportunity, Dr. Ho introduction, and so today I'm going to talk about some tough targets, how we really tackle those multi-pass transmembrane protein to develop therapeutic antibodies. Actually, why we choose this area, it is a kind of fulfill my dream during my research time. When I did my postdoc research in Hopkins, I work on ion channel. I still remember when I purified the protein. The first day I purify, and the next day, then I store in -80. We use a detergent. Next day we saw from -80, all the protein aggregate again. I lost the function, lost the whole week of effort. That's quite a struggle. And when I built up my own company, I said, that's the best time. Use my own money and the investor money to fuel my dream and solve the problem in the field. Yeah. So let me first give everybody a quick overview about DIMA Biotech. We are a biotech company focused on hit to lead stage of the therapeutic antibody discovery, and mostly focus on preclinical and especially before CMC stage discovery. And so in today's talk, I will be focused on three parts of our technology platform. The first part is definitely the most focused on how we purify and characterize the multi-pass functional membrane proteins. And the second part is a very brief introduction about our single B platform, how we really clone the IgG gene from the B cell instead of doing the hybridoma fusion. And the third part is a very quick introduction about our mammalian display technology for antibody engineering, humanization, affinity maturation. And the second portion, I will use two of the cases to show how we utilize the platform built up to screen the biologics. And one of the target we work with CAR-T company already moved to the clinical stage with those 17 patients in one center, another 4 patients in another center show very promising clinical efficacy. Is a GPCR target. Lastly, and most importantly, we are an antibody company, so we have a very unique off-the-shelf lead molecule out-license business model. We guarantee 0 upfront, 0 waiting time, and so 0 risk for you to speed up your discovery process. I mean, how we really can collaborate, work together. And the first part is about our DIMPRO transmembrane protein technology. And so there's a different technology in the past people develop. The most well-known is like a detergent approach. People use that for decades for the protein purification for multi-pass transmembrane protein because the transmembrane part is extremely hydrophobic. So when you purify in the aqueous solution, they tend to stick together from a big aggregate, lost the function of the 3-dimensional structure. And then these two are the NanoDisc. The traditional NanoDisc we talk about, the MSP-based NanoDisc and the synthetic polymer SMA-based NanoDisc. And we also developed another NanoDisc approach called a peptide NanoDisc. Also people call it PEGS. And everybody know, I mean, I don't have to say more, to develop therapeutic functional antibody, the immunogen is the key. So without a functional protein, it's very hard to develop whatever in the downstream. And for biologics, antibody-based, like the drug, 90% of them are the transmembrane proteins. Based on the complexity of the transmembrane protein, you have 2 major categories. It is the first one, it is a single transmembrane protein. Another one is a multi-span transmembrane protein, like this GPCR. So you have 7 transmembrane parts. For single transmembrane protein, there's already well-established, like an easy solution. People just take the extracellular part, express as a aqueous soluble version, and just like a bacterial protein expressed in the mammalian cell and form a functional domain. And for the multipass, if you just take off the extracellular domain, cut it out, then it's very hard to mimic the native structure. The antibody you generate may not recognize the native conformation. So how to do this? In the past, people use whole cell membrane prep to do this, but there's a lot of challenge because there's a lot of background clones during the screening. How to solve this issue? We developed a couple of different, as I mentioned, different technology for this. So that's what I'm going to talk about. And membrane protein, everybody know, I don't have to say more, it's highly hydrophobic, and also the yield is really low. A lot of case, we do like a 1 liter of the cell, suspension cell, transfection and purification. In certain, sometimes we only get like a 50 to less than 100 microgram of the purified membrane protein. Definitely, they're also target dependent. There's certain membrane protein you get a little bit more, but this is a very challenging part because they only distribute on the membrane part, and the membrane is only very small portion of the whole cell in terms of the volume. And the second part is that they tend to be very unstable during the storage. And so everybody know, GPCR is a good druggable category for the protein. Out of 800 GPCR proteins, more than 300 of them already have small molecule drugs. But the FDA approved biologics target on GPCR, only 3 of them. What is the reason why such a big druggable target group of protein, you only have 3 of them? The challenge is not because of the antibody technology, it is the immunogen preparation. Yeah, how to solve this issue? So people try to really solve this issue by, detour by using different approach. So one of the common approach people use is VLP or exosome to prepare the membrane vesicles. This will exactly mimic the native structure of the protein. They secrete from a cell. You can use that as an immunogen. It's a good approach. But one of the challenges is the yield is really low because and also you introduce other non-relevant protein like GAG, lentivirus. It's highly immunogenic. Then you will also get antibody for those. And so this is a 2 case. Claudin 18.2, we use this as an example. So this is after we made VLP, we run the gel. You can see most of the protein is non-relevant host cell membrane scaffold protein. And your target protein is very little, minuscule. And Claudin 18.2, the yield, actually the production yield is relatively higher compared to all the membrane protein. Even that, you don't see the distinct band, only can detectable by the Western. And the exosome is the same. In this case, if you use that to do the immunization, you can think about it. Most of the antibody might be just attack, against your non-relevant membrane proteins. Yeah. And also it's very hard to quantify each batch because it's a physiological process secreted from the membrane. And also when you do other analytic analysis also is very hard, like SPR. And so how to solve this issue? Whether we use this NanoDisc approach. So this is the one I just draw here. Yeah. And so comparing to detergent approach, what is the benefit with the NanoDisc? So detergent, everybody know, is you basically wrap around the transmembrane part, hydrophobic part, with this detergent, which is amphipathic. One side hydrophilic, another hydrophobic, like we use for the washing the clothes. Yeah, to get rid of all the lipid. Yeah, and so no lipid involved. But the NanoDisc is a different scenario. So what they do is they use the MSP protein or synthetic polymer they have the similar property. One side hydrophobic, another side hydrophilic. So they can circularize around the lipid bilayer, form a small disc, 5 to 15 nanometer in diameter. Also in the center, still you have this membrane protein reside in the lipid bilayer, have the native structure, most native conformation. And so by this way, it's a nano disc. You can purify by the chromatography, either affinity purification or other approach. And so here we use an example. We purify Claudin 18.2 by traditional detergent, and we develop this synthetic NanoDisc approach, and with this peptide NanoDisc approach. So you can see, as everybody know, I mean, when you use a detergent purify, because some of the highly hydrophobic protein also extracted, so they tend to bind non-specific to the column. So you have much more, non-specific binding. But for the synthetic NanoDisc, you can see here, they are more pure, just one round of affinity purification. And this is a peptide NanoDisc. You have this protein, and also you have this peptide, which stabilize the transmembrane part. Yeah. And so this is, this Claudin 18.2 form a monomer, dimer, and a trimer. So it's normal 3 bands. And if you use, so if you do immunization, how it works. So when we did a trial test, so basically these 2 mice, we use VLP, Claudin 18.2. They gave a slight immune response. So this is a different titer. And when we use our synthetic NanoDisc, give a very strong immune response. Yeah, so this is because you can imagine, because you inject the pure protein, that's very logic. And so we also combine mRNA like immunization and a whole cell immunization with NanoDisc, because mRNA immunization sometimes depend on the target. The serum titer is a little bit challenge. So we combine that with a NanoDisc here, and so you can boost up immediately with on the target. And this is because the NanoDisc, everybody complains, is very expensive. So this is the way you can compromise in the meantime, also achieve your goal. Yeah. And since we have synthetic NanoDisc with the lipid bilayer and very native structure, why we develop this peptide NanoDisc with the peptide wraparound from the NanoDisc? What's the reason for this? Why? So actually, when we do our experiment, we have an urgent need to really do cell-based assay, especially for the B cell screening for the FACS binding to the multipass transmembrane proteins. That's one of the challenges. Is synthetic base, like this membrane embedded NanoDisc, they have this property called the nanotransfer. So when you incubate this NanoDisc with the live cell, they will non-specifically fuse with the membrane, cause non-specific binding. So the binding is not dependent on the target anymore. So then we try to figure out use a detergent, but detergent has some detrimental effect to the live cell. And the peptide NanoDisc, the good thing is it will wrap around with the peptide. It's a whole protein base. So you can use that for the B cell identification and the other cell-based assay. So that's what we develop. And this is the example. We make like a Claudin 18.2 CAR, and we transduct to the T cell, and we can quantify what's the transduction efficiency by using this Claudin 18.2 peptide NanoDisc. And we validate also with a G4S antibody, because you can also use another approach. So even though DIMA is a therapeutic antibody discovery company, we pay quite a tremendous attention on the functional protein, because we truly believe without a good functional protein, you develop therapeutic antibodies, it is a totally waste of time So then I was switch gear about our single B technology. For this is our process. So basically we immunize with the animal, then we from the PBMC and the spinal side, we isolate memory B cells, which has mature antibody and also transmembrane domain, and like a BCR. Then we cultivate this B cell in our proprietary single B cell cultivation media with like a feeder cell and the growth factors. They can proliferate. I mean, then we clone the gene. And so after we identify the lead, the second thing is how we make them as a humanized antibody and improve the affinity by affinity maturation, antibody engineering. So we develop our own DIMA mammalian cell display technology. So this is the figure elucidate how we construct this. Basically, the whole antibody will display on the cell surface. We add a transmembrane domain like a BCR, and also we add a EGFP to monitor the antibody expression and the trafficking process, the efficiency. And also you can use this to display the whole cell. I mean, mammalian cell or CHO cell, I mean, HEC cell or CHO cell, you can do another round of like a panning screening. You get like the humanized or affinity matured antibody. By this approach, why we do this tough way? Because by this approach, we smartly utilize the mammalian cell internal quality control system. So basically, in the cell, the proteasome and the lysosome is a good your working partner. So they can detect if your protein has any small defects in terms of the three-dimensional structure. They will be degraded during the synthesis at the ER by proteasome. If they manage to go to the cell surface, they will be degraded by the lysosome. So using FACS sorter, you can always identify those molecules with a good developability and also the affinity. And so here I'm going to show two of the examples. And so first one, it is at 18.2. We develop an ECL1 loop specific therapeutic antibody. And so everybody know, I mean, 18.2 and 18.1 come from the same gene through alternative splicing. The only difference is the ECL1 loop. So you want to develop an antibody only specifically recognize the ECL1, but not bind to Claudin 18.1, not Claudin 18.2. So this is the only 9 amino acid difference. How to really tackle this challenge problem? So we utilize this synthetic nanodisc and peptide nanodisc. So first, we collect the PBMC or splenocyte. Then we first get rid of any reactive to Claudin 18.1, use a peptide nanodisc. So just use a MAX to get rid of them. Then we go through another round of this MAX and enrich those 18.2 specific binders, and we cultivate the B cell clone the gene. And after humanization, and we made the CAR construct. And so here, this purple color, it is like a carcinogens in clinical stage, phase 2 clinical stage CAR Ts. And so as a benchmark, and these 4 are our humanized CAR T. You can see we did like an in vitro tumor cell stable cell line killing. So K562, 18.2 show very specific killing, not on this 18.1. And in terms of the efficacy, it's comparable, or some of them might even better. And so another case, it is a GPCR target, it's a GPRC5D, it's a multiple myeloma target. And so this project we work with Guangzhou Biologics, it's a CAR T company. So they have a CMC and a clinical team. And so I mean, GPRC5D right now is the hardest target for multiple myeloma treatment, especially for BCMA resistant patients. And their bispecific antibody 2023 J&J's tacrolitamab get approved. And so BMS is doing the phase 2 clinical trial with CAR T, GPRC5D, and there are also tri-specific antibody and the dual CAR T also in the clinical stage. And so after we screen all the antibody, we identify 9 clones and humanized clones. We compare with the BMS MCAR H109. So here's the BMS one. So it shows several of clones showing much stronger binding in terms of the cell-based FACS assay. And we did an animal study also. We chose 3 of them. We did an animal study and it shows good in different like a dosage of the infusion, and 3 of them showing very good tumor suppression. And we choose G05 and G07. We work with Yunnan Hospital, Cancer Hospital. We dose 17 patients. And with Tianjin Hematology Research Institute, we dose 4 patients with G05 and G07. And this is the clinical data, and it shows out of 17 patients, and our CR rate reached to 65%. And for the GPRC5D positive patients, we achieved 100% ORR. And so that is basically the technology for the very brief description, I described very briefly about our technology. Here's our products and our off-the-shelf lead molecules. Currently, we already developed more than 500 druggable target, 5,000 molecules against 500 druggable targets. All these have been done with in vitro assay validation to different stages. Some of the is just like a FACS binders. So some of them we did a humanization and also like a functional evaluation for different modality. And so why we can do this? Because we dissect the whole very complex monoclonal development approach into a modularized pipeline approach. So we can, in 1 week, in our team, we can almost do 6 targets in parallel to push all together. So that's why in the past 8 years, we have achieved all this. For each, try to really improve the diversity for each target. For each target, those important target, we immunize sometimes 10 animals to get a more coverage of the diversity and also different species. We do rabbit, we do mouse, and we also do VHH. So if you have any interest on your project, we can work, and since it's off the shelf, we guarantee it is no upfront because it's already in this industry everybody respect intellectual property. So we sign the MTA, we send the protein to you, you validate, you don't pay anything. You validate, works, then even some of our partner, they have their internal molecule or with the other CRO molecule, doesn't matter. You don't have any risk. You validate in parallel, see how our molecule comparing to your molecule, because if yours is better, you don't have to, you just walk away. But if ours better, you will really mitigate the risk for the downstream because the real cost is on the clinical stage. If you choose, and I would say, if you want to select a best, champion for the Olympic, best from the baby side. So if you choose a round baby, eyesight is not good. You do like the shooting test, it's not good. So basically, we help you and much more economic, efficient way to really speed up your discovery process. Yeah. And so here we have a booth downstairs at 823. You're welcome to come over. And we do have this, I think my colleague sent this flyer to you. You're welcome to ask any questions. Yeah, thank you. Yeah, if you have any question, I will take.
So any questions? So your clients are mostly industry, not the academic labs?
We do have quite academic lab. They do translational study. So they work with us, and then we can do co-development. So you pay little, but we can get the upside in the future. Yeah, yeah. We are very open. Yeah, in terms of business term also.
Go
So
ahead.
thank you so much. Really great talk. So my question has to do with when we generate nanodiscs, we know that the polymer we use and the buffer turning it in can significantly affect the size and the properties of the nanodisc. Have you done any studies into which has the most physiologically relevant like surface tension?
Yeah, that's a very good question. Actually, synthetic polymer base, like SMA base, there's a lot of different, people have different polymers with different property of the transmembrane protein. It might affect, the biophysical property of the membrane, the curve, and we do notice certain protein, they might have functional effects. But this has to be, that's why every protein, transmembrane protein, after we prepare, we always do functional evaluation. So if a GPCR has an native ligand, we do a ligand binding assay. So if you don't have the, often GPCR like a receptor, we'll see whether there's any clinical stage therapeutic antibody. We will synthesize and validate our protein before we immunize the animal. Yeah, so that's what we do. And we do see there's certain case, and we do. Do have a slide. Certain case is not a synthetic polymer based with the lipid membrane. They are more functional. We see peptide nanodisc more functional. So maybe I can show you this one of the data here. So I do have a lot here, but I didn't show here. See here, CD19. So the orange, it is a peptide nanodisc. When we test bind to the clinical stage therapeutic antibody, it works. But the synthetic polymer base doesn't work. So you really have to not just make the protein appear as a protein. That's called a peptide. So you have to functional validate that is works.
Do you have a list of all validated products on your list or in your booth?
We do have the list of all 500 these druggable targets with 5,000 molecules for all different applications. You are welcome to come over. We can send you the list. Yeah.
Some of the epitope space on a protein is dependent on either conformation or oligomerization. I'm curious whether you're able to model those protein-protein kind of interactions in the nanodisc so that you can capture the native conformation and epitope space.
Yeah, that's a very good question. So basically, there's always people say when you purify the native and we overexpress in the H293 cells, definitely there will be some associated proteins because if you get rid of associated protein, they're not stable. So it's a balance. But in most of the cases, you overexpress, if the protein can be stable in as like a tetramer or monomer, and in itself, we show very distinct band. But sometimes we do see multiple band at the bottom, and those could be associated protein, but they are much less. And also, when you see all this, we validate with the like SDS page. If you go to our website, we for the more than 300, 400 this membrane protein we have off the shelf, we also validate by the SDS page. You can see multiple bands. So actually not means this protein not pure. It is because the transmembrane protein, they are highly hydrophobic. Even during the SDS page running, they form aggregate cross-link. When you do the same protein, you run the SEC, the highly homogeneous, just a single peak. So, and I can't rule out there might be associated proteins in this case. And we also see certain, hetero, like a beta-adrenergic receptor is a pentamer. You also can form a complex on the nanodisc.
Yeah, so the question more so is for proteins where there is a known like hetero-dimer, can you express both so that you capture that?
You can.
Okay.
And we use bacular virus. So co-infect with the backman. So that's you can control the stoichiometry of the assembly. Yeah.
Thanks.
The last question.
I saw you immunize with the mRNA and the NanoDisc as well. And then there are 3 different flavor of the NanoDisc scaffold-wise. Is there any toxicity to the mouse, or is there any preferred flavor among the SMA, MSP, or peptides?
So you can use SMA based on NanoDisc for the immunization, and also you can use peptide NanoDisc. Normally what we do is we use one polymer base to do the immunization, and we use a peptide NanoDisc for the screening. And mRNA and also you just asked another question is whether we tried all different animals. We tried, yeah. So we tried with rabbit, we tried with mouse, and we also tried with alpaca.
So you don't have any toxicity for that?
Yeah, it all works, yeah. And also,
very good. Thank you.
Yeah, sorry.
Thank you, Dr. Ma, for very exciting lunch presentations.